Welcome to Cell Biology for Pharmacy. This video is the fourth video in a series on the cell cycle. In this video, we will dive into the details of mitosis and cytokinesis, which make up the M phase of the cell cycle. Let us begin. Right, so as we've seen before, division of the cell into two daughter cells occurs at the M phase of the cell cycle. This phase consists of two parts. The first is division of the nucleus, called mitosis, and the second is division of the cytoplasm, called cytokinesis. As you can see here, mitosis is further divided into 5 stages, followed by cytokinesis. We will discuss each of these stages in detail in this video. If you recall from the previous video, only cells that have successfully duplicated their chromosomes and organelles may enter the mitosis phase. Cells that have failed in their DNA replication or are delayed because of other reasons cannot yet enter the mitosis phase. Now, for cells that can enter the mitosis phase, each of their chromosomes have been duplicated and consist of two identical sister chromatids joined together at the centromere, as you can see from the image. During mitosis, the two sister chromatids are split apart, and each chromatid is pulled apart to opposite ends of the cell. One of the most important structures that are involved in this process is the mitotic spindle, which we will also discuss in this video. The mitotic spindle is made up of microtubules, which are cylindrical protein structures that make up the cytoskeleton. Here on the left, you can see the molecular structure of a microtubule, which is a polymer of tubulin proteins. In animal cells, the mitotic spindle is generated by a microtubule organizing center called the centrosome, as seen on the right. Microtubules are generated and grow in all directions from the centrosome. Now, before the cell even enters the M phase, it's critical that the centrosome is also duplicated during the interphase, along with all of the cell's DNA. Duplication of the centrosome typically occurs at the same time as DNA replication, which is during the S phase. In the first phase of mitosis, called the prophase, two main events occur, inside and outside the nucleus. Inside the nucleus, the duplicated chromosomes start to condense into sister chromatids. DNA inside the nucleus are usually not stored in the form of chromosomes in order to allow gene transcription and other processes to occur. It's in the prophase that the DNA starts to condense into chromosomes. Outside the nucleus, the mitotic spindle is formed. In this light micrograph you can see on the right, you can see that the microsomes in red starting to take shape, and the dynamic and random growth of the spindle microtubules in green. As we've seen before, the mitotic spindle microtubules are formed from the centrosomes. At first, new microtubules grow in all directions from the two centrosomes. Now, microtubules are inherently unstable, which means that they tend to easily break down. Therefore, some of the microtubules that were rapidly growing outward will start to shorten instead, as you can see from the red arrows here. Some of the microtubules growing from one centrosome will start to interact with those from the other centrosome, and when this interaction occurs, motoproteins, these black dots here, will interact, and this stabilizes the microtubules and prevents them from breaking down. These microtubules from the two centrosomes that interact with each other form the mitotic spindle, and the centrosomes are now called spindle poles. The next phase is called prometaphase. The start of prometaphase is signaled by the abrupt breakdown of the nuclear envelope, also called the nuclear membrane. Before the nuclear envelope disintegrates, the growing spindle microtubules were separated from the chromosomes. The microtubules were outside the nucleus, and the chromosomes were inside the nucleus. Now that the nuclear membrane is no more, the spindle now has access to the duplicated chromosomes. Now these spindle microtubules that are growing randomly from the centrosomes will eventually encounter their chromosomes, and once this happens, the spindle will capture the chromosome at the kinetochores. So, what are kinetochores? Kinetochores are protein structures located at the centromeres of each condensed chromosome. They are assembled at the later stages of prophase. Each duplicated chromosome has two kinetochores, one on each sister chromatid, which face in opposite directions, as you can see here in red. The spindle microtubules that capture the chromosome links the sister chromatids to the different spindle poles. This one on the right links this sister chromatid to the pole on the right, and the spindle microtubules on the left links this sister chromatid to the pole on the left. In this fluorescent micrograph of a duplicated chromosome, you can see the DNA stained with a fluorescent dye, showing a brown color, and the kinetochores stained in red. The third phase is called the metaphase. As we've just seen, in the previous phase, the chromosomes are attached to the mitotic spindle. They are now moving around erratically, as if they are being pulled in different random directions by the spindle. However, soon all of the chromosomes will eventually line up at the equator, which is halfway between the two spindle poles, forming what is called the metaphase plate. This signals the beginning of the metaphase. At the equator of the spindle, the chromosomes do not stay still. They continue to oscillate back and forth as if there is a tug of war between the two spindle poles, and the chromosomes are constantly being pulled toward each pole. Experiments have shown that if a laser beam is used to break one of the spindle microtubules attached to a chromosome, the chromosome will immediately move towards the spindle pole that it's still attached to. Other experiments have also shown that if the bond holding the two sister chromatids together are broken, they will immediately move apart towards each spindle pole. These experiments show that the duplicated chromosomes don't simply move towards the equator and then stay still, but are held under tension by the two spindle poles. Think of it as the two poles are fighting each other, trying to pull the chromosomes in their direction. Another thing to know is that the spindle microtubules are classified into three different classes. Microtubules that attach the chromosomes to the spindle poles are called kinetochore microtubules. Microtubules that interact with the other microtubules from the opposite poles are called interpolar microtubules. And microtubules that don't interact with either the chromosomes or microtubules from other spindle pole are called astral microtubules. The next phase, called anaphase, starts abruptly with the breaking of the link that holds the two sister chromatids together. Because the sister chromatids are held at a state of tension due to the pull from opposite spindle poles, the breaking of the link causes the two sister chromatids, now each considered chromosomes, to move toward opposite spindle poles. Here you can see the transition between metaphase on the left and anaphase on the right. The sister chromatids, which are stained blue, suddenly separate and the separated chromosomes move toward opposite poles. They both move at the same speed at about 1 micrometer per minute. This movement is made possible by two separate mechanisms called anaphase A and anaphase B, which depend on different parts of the mitotic spindle and occur simultaneously. In anaphase A, the kinetochore microtubules depolymerize and shorten, generating a pull force at the kinetochore, and the daughter chromosomes are pulled toward opposite poles. In anaphase B, the two spindle poles are pushed apart due to two things. The first is that the interpolar microtubules elongates and slides past one another, pushing the poles apart. And the second is that the astral microtubules that point outward are pulled towards the cell cortex or the cell membrane, hence pulling the two poles apart. It's the combination of these pulling and pushing forces which drive the movement of the chromosomes and the separation of the two spindle poles. The last phase of mitosis is the telophase. Once anaphase is completed, the daughter chromosomes are grouped at the spindle poles at opposite ends of the cell. During telophase, the mitotic spindle is broken down and disassembles, and the nuclear envelope starts to form around each group of chromosomes to form the two daughter nuclei. Once the nuclear envelope has reformed, the chromosomes start to decondense, and once that's complete, the two nuclei are fully functioning and can perform DNA transcription once again. This marks the end of mitosis. All that's left for the cell to do is to separate physically into two daughter cells in a process called cytokinesis. Cytokinesis is the process in which the cytoplasm is divided into two, as you can see in this picture. At the end of telophase, a contractile ring has started to form around the middle of the cell, forming a ridge around the cell. This contractile ring becomes smaller and smaller and eventually pinches the cell into two daughter cells. The contractile ring is formed by actin-myosin filaments. Here are two electron micrographs in which you can see a ridge forming around a dividing cell. Once the cell has been divided into two daughter cells, this marks the end of the M phase, and each of the daughter cells can enter the G1 phase. That's the end of this video. Next, we will discuss what happens when cell division starts to go wrong. Thank you.